Welcome, everybody, to this webinar on biotechnologies beyond conventional containment. My name is Richard Murray, and I'm the Thomas and Doris Everhart Professor of Control and Dynamical Systems and Bioengineering at Caltech, and one of the organizers of the Lynn Policy Center's activities in biotechnology. Thank you for joining us today. We had well over 100 people registered for today's event, and we're looking forward to a great session. Let me first say a few words about the sponsor for today's webinar. The Lynn Center for Science, Society, and Policy, LCSSP, or Lynn Policy Center, was established in 2023 and aims to serve as a national forum for research, debate, and dissemination of knowledge on topics of interest at the intersection of science and society. The center's mission is twofold: to advance understanding and education on contemporary science policy, and to provide data-driven guidance for policymakers and other stakeholders by leveraging Caltech's distinct scientific expertise. In the biopolicy area, we ran a workshop in February on engineered microbes for environmental release, or EMERs, that brought together researchers, technologists, policymakers, and others. And in June, we issued a report with some policy recommendations coming out of our workshop, which you can find on our website. Today, the Lynn Policy Center is pleased to be able to host a webinar that brings together some of the coordinators from the recent Spirit of Asilomar summit that took place in late February, and that I'll describe in a bit more detail in a moment. First, a few logistics for our webinar this afternoon. The video and audio portions of this meeting are being recorded and will be posted on the Lynn Policy Center website at a later date. You can ask questions at any time using the Q&A function in Zoom. Please note that if you ask a question today, your name may be captured and other members of the audience may wish to follow up with you after the webinar, including reporters who have registered for this event. Feel free to like questions that have been posed to elevate them for priority placement for posing to the panelists. I'll be monitoring the questions and passing them on to panelists in the second half of our webinar. So the plan for today is that we'll spend about 45 minutes having each of our panelists say a little bit about their experiences and their work at the Spirit of Asilomar Summit. And then we'll open it up for questions for about 45 minutes. We'll only go about an hour and a half for questions from the audience and discussion. So with that, let me give a little bit of background and context for today's webinar. So the Spirit of Asilomar Summit was called the Spirit of Asilomar and the Future of Biotechnology. As you see, it was on the 23rd through 26th of February, so just about 6 weeks ago. And we're going to talk today about one of the themes within that webinar. So first, let me just say a little about the summit and how it was organized and who was there, and then I'll talk about the themes. So the Spirit of Asilomar Summit brought together about 300 scientists, conservationists, bioethicists, lawyers, former government officials, national security experts, journalists, both internationally and a set of next generation leaders, or NGLs, at the historic Asilomar site. Those of you who follow biotechnology know that this is where the first Asilomar Summit 50 years ago, started on the 24th of February, 1975, was held on recombinant DNA technology. This summit was intended to bring together a broad group of people, including various points of view, both nationally within the US, but very much so internationally, and both people who'd had a lot of experience in this area, as well as people who were at the early parts of their careers. The way the summit was organized was into a bunch of working groups, which I'll describe in a minute, and a bunch of themes, which I'll describe in a minute, and then into a bunch of working groups underneath those themes. Over the course of 3.5 days, we essentially talked and debated about things that needed to be done in areas related to future biotechnology. And the plan going forward is that various groups of people have put together statements and reports and entreaties. We use the general term entreaties. Draft versions of these were due on the 14th of April, and we are right in the middle of a feedback period from the summit participants, but also from the public in the sense that the things you hear here today are things that are in the entreaties, and we welcome your feedback on it. On the 28th of April, so in a few weeks, we will, so on the 14th of April, we'll get all of that feedback, including feedback from these groups, we hope. The 28th of April, we have to do revisions of those entreaties, taking into account that feedback. And then there's a process by which people can sign up to endorse different entreaties, and those entreaties that receive at least 10% of the participants, so 30 endorsements, will eventually be posted on the Asilomar website in late May. So that's kind of the idea here, was to have a discussion, to use that discussion, have more discussions, and then eventually to try and come out with some outputs from this. And the entreaties are, as you will see when they come out, in a variety of different forms, and you'll see a little bit here today as you hear about them. So there were 5 themes that were used to organize the discussions. Those themes are shown here: artificial intelligence and biotechnology. The theme that we're going to talk about primarily today, biotechnologies beyond conventional containment. And I sometimes just say beyond containment for short. Another on framing biotechnology's future. So how do we think about biotechnology generally, the bioeconomy, different ways of kind of equity and access within the bioeconomy or within biotechnology generally, things of that sort. A fourth on pathogens, research, and biological weapons, and the last on synthetic cells, including mirror biology. So each of those themes had a number of working groups. Again, here today, John Marken and I, who's one of the speakers today, were the co-leads for the Biotechnologies Beyond Conventional Containment theme. We had 5 working groups within that theme. Those 5 working groups were one that started on environmental safety levels and turned into something that was a little bit more about governance frameworks. Another on public infrastructure to support biotechnologies beyond conventional containment. Another on design guidelines, and a fourth on the intersection of ecology and conservation with biotechnology. There was a fifth on ethical, legal, and social implications. This was not really a separate working group, but rather a separate area that we recognized was very important and that we sort of distributed across these areas as well as had them kind of meet separately to kind of talk and coordinate about how to address ethical, legal, social implications across that. All right, so this was sort of a little bit about the way the summit was organized. Again, 5 themes. Each theme had, let's say, 3 to 5 working groups. And those working groups, 300 people, right, sort of divided down into groupings of, anywhere from 8 to 24 people in a room, maybe even more in some of them, talking about these topics over the course of 3.5 days with various, plenary sessions and other things in the middle, as you might imagine. So in the area of beyond containment, I want to start by just saying a little bit about what beyond containment is, and then I'll turn it over to the panelists to talk about sort of their views on these things. So what do we mean when we talk about beyond containment? So you can think about 3 different sort of situations. One is a factory situation where you have a closed environment. You're putting something in a fermenter, for example, or a factory of some sort, that's really closed to the external environment, and you can control what happens with that biotechnology. Typically, you would do something where you would, treat any biological material before releasing it out of the environment through a waste stream or something else. So a very kind of closed environment sort of a situation. You can have a farm, and a farm, of course, is something that's more open to the external environment, but still something that we can contain in various ways. We have geographic containment. We know how far things spread. We can think about if we're going to have a genetically engineered crop, for example, the different mechanisms by which those we might have gene flow or other things. So partially controlled. And then you can have field release. So something where you really are going to release engineered or biotechnology engineered organisms, perhaps into the field. And so that interface with the environment's very large, and your ability to control what happens afterwards may be less. And so in 1975, they really focused on sort of the containment part. What do we do in the lab? How do we contain things in the lab as we're starting to understand more about recombinant DNA and being careful about how we do things there? In the farm situation, of course, this is widely used in the context of GMOs and other things around the world. And so it's at least easy to conceptualize. We know we've got a farm. It's got some genetically engineered microbes or plants or maybe animals. And it's easy to conceptualize. We want to keep it there. But it's a little bit hard to implement, right? How do you constrain things to sit in that environment? But really, we're interested in biotechnologies beyond conventional containment. So this third category, where it's not even clear what you mean by containment, because the whole point is you're trying to maybe affect the environment. Maybe that's for conservation reasons. Maybe it's for remediation of some sort. And so how do we think about that? And so in 1975, we sort of started on the left, and today we're very much looking at biotechnologies that might impact things on the right. And we have to think about, do we want to employ those biotechnologies? What benefits do they provide? What are the alternatives to doing those things? So that's the way that we sort of think about this particular theme. And I'll say this figure and this idea came out of a review paper by Jones, Marken, and Silver in 2024 that you can look at for more. So with that, let me hand things over to the panelists to kind of give their perspectives on their parts and what they did both at Asilomar and within the Beyond Containment theme and within some of the working groups they participated in. And I'll ask each of the panelists to briefly introduce themselves at the start, just so you know who's speaking. Okay, and so we'll start with John Marken.
Hello, thank you, Richard. So hi, everybody. I'm John Marken. I'm a postdoctoral researcher at Caltech. And as Richard mentioned, I was one of the co-organizers of the Biotechnologies Beyond Conventional Containment theme at the Spirit of Asilomar meeting. And I want to talk today about one of the working groups at the meeting, specifically that one that was focused on an environmental safety level framework. So just to give some context, evaluating the risks and benefits associated with the use of any particular biotechnology is a very complex and challenging problem. And so what can be useful to help aid in that process is to develop evaluative frameworks that help make those decisions simpler and also more consistent across different cases by providing an overarching framework to guide these assessment decisions. So one prominent example is the biosafety level framework, which is used by many countries to categorize biological agents into 4 different categories of risk. And the biosafety level framework provides guidance on what sort of precautions need to be taken before the use of agents in each different category can be allowed for activities within the laboratory. So at the Spirit of Asilomar meeting, one of the working groups convened to ask, can there be an evaluative framework that we can come up with that would be similar to the biosafety level framework in the sense that it provides this kind of overarching guidance about how to think about evaluating and assessing the potential risks and benefits of a particular use case for a beyond containment biotechnology. So if you could build the slide, Richard. So a couple key conclusions that came out of these discussions, and the major point is that biotechnologies beyond conventional containment are fundamentally different from conventional contained biotechnology from the point of view of generating frameworks for thinking about their evaluation. So for one, beyond containment biotechnologies, because they're going to be released into the open environment and interact with that environment, will have multidimensional impacts. So those impacts could be ecological, they could be economical, they could relate to human health, they could relate to human cultures and values. All of these types of dimensions will have impacts from the use of these biotechnologies out in the open environment. And similarly, these impacts will be multi-scale. So you'll have impacts at the local scale, perhaps the regional scale, and potentially even the global scale. And so these frameworks need to be able to evaluate these multidimensional, multi-scale impacts when considering the use of a beyond containment biotechnology. Another point where these technologies differ from conventional biotechnology is the fact that every use case associated with releasing a biotechnology into the environment will be linked to a specific goal. Because the notion of releasing engineered organisms into the environment is not something that we can or we should or do take lightly, every single potential use case is going to be associated with a concrete set of specific goals and objectives. And so, unlike with conventional biotechnology, where almost all use cases share the same first order goal, which is don't let the biology escape from the laboratory, in the case of biotechnologies beyond conventional containment, the first order goal may differ from case to case. And so while that does provide a standard on which to frame the evaluation, the fact that that standard may differ from case to case will also make it challenging to develop a framework for evaluating these things. And finally, it's not just the standards and goals that will change, but also the specific properties of the use case. Is it going to be released in a large scale setting, a small scale setting? What sort of environment, terrestrial, aquatic, all these things are going to factor into the evaluation process. And the framework will need to be flexible enough to accommodate these things. So if you could build one more time, Richard. And so the major conclusion that came out of this is that evaluation frameworks for biotechnologies beyond conventional containment need to be developed hand in hand with frameworks for the governance of these same biotechnologies. And furthermore, these frameworks, both for evaluation and for governance, are going to have to deal with unique properties of beyond containment biotechnologies in such a way that existing frameworks for conventional biotechnology are not necessarily going to be a good or useful reference. So if you go to the next slide, one example of what this integration between evaluation frameworks and governance frameworks might look like is shown in this diagram here. And so a couple of things I want to point out. One, you can see that the concrete objective is kind of saliently captured as the frame of reference across all steps of evaluation. And so that kind of speaks to the fact that every use of a biotechnology beyond conventional containment must be in service of this concrete goal. And similarly, we have represented here an iterative design process that incorporates stakeholder input from affected stakeholders and communities early on in the process. And so the hope is that this type of framework encourages the development and iteration of the technology, even in terms of the actual method of implementation. So it may be that the final implementation may not even be a biotechnology solution at all. It may be a non-biotechnological solution if it turns out through going through this process that a non-biotechnology solution turned out to be the best way to meet these objectives and also the other priorities of the stakeholders involved. And so once again, this is just a rough, vague conceptual outline of what this sort of thing might look like. But we recognize that we're seeing that, you know, the types of properties that these beyond containment biotechnologies have are going to be better served by these more flexible dynamics forms of governance and evaluation, rather than more rigid kind of category-like structures that are useful in conventional biotechnology. And so just as a final build, one more build, please. So there are still many, many open questions associated with what evaluative and governance frameworks for these technologies are going to look like. Some major questions, once again, include how do you develop a framework that can be flexible enough to accommodate all of the different cases? And also the fact that the impacts are going to be multi-scale and multi-dimensional is going to mean that it's not necessarily clear who the stakeholders, in quotes, are, and what do we do about the fact that different groups of stakeholders are going to have different values and different interests that may conflict in terms of going through this evaluative process for the technology. And so, and finally, how do we learn from other fields? So I mentioned previously that it's looking back at frameworks for conventional biotechnology are not going to be the appropriate reference frame. And so what is the appropriate reference frame? What are other situations and other types of technologies that involve this question of multi-scale, multi-dimensional impacts, for example, and what can we learn from them? So once again, this is a lot of challenges and a very difficult problem to kind of develop these frameworks. But I think also the fact that, you know, these types of biotechnologies are really emerging prevents us the opportunity to be flexible and dynamic with the way that we think about governance and the way that we think about evaluation. So hopefully, I think one shred of hope that came out of the discussions is that even though it's a very challenging process to come up with these frameworks, it also can be an opportunity to explore new types of frameworks that may be more effective down the line because we have sort of an open playing field to think about how to experiment with these sorts of approaches. So yeah, thank you.
Great. Thanks, John. Okay, next up, Andrea.
Hello, everyone. My name is Andrea Hodgson. I am Director of Biosciences at Schmidt Sciences, and I'm reporting out on behalf of the Public Infrastructure Working Group. And we had the opportunity to really envision how we might better ask and answer questions around the risk and benefit of the release of either engineered organisms, and actually, in our case, we had a really broad sort of approach to the definition of what public infrastructure would be, as well as release and introduction. So we had the conversation in the context of recognizing that the solutions here for engineered microorganisms or engineered organisms in general would also be applicable to the release of any type of non-native species into a new environment. And so we approached the definition of public infrastructure super broadly and recognize that there were a number of challenges that we have when it comes to thinking about beyond biocontainment of biotechnologies, and then recognize that there are a number of core elements or facets of public infrastructure that were worthy of discussion. And so the slide here outlines two of the major challenges we face when we think about the introductions of organisms into environments, whether they are non-native organisms into a new environment or engineered organisms. We really lack an ability to test the full complexity of how the environment will impact that organism and the potential for that organism to spread beyond its intended release area. Even in the context of some of these technologies where you might want it to spread, we really lack the ability to accurately model and understand what those interactions will do, how that will impact the organism, and how it will impact the environment itself. And we also don't have any comprehensive sort of public accounting of all of the release events. And this is, again, in the context of both engineered and non-engineered organisms. We don't have a central sort of aggregated sense of information around where have there been release events. We certainly have an understanding of what permits have been applied for, but that doesn't always translate to a field release or a trial, a field trial. And it certainly is not, there's no sort of mass aggregation of information around how often approved organisms have been introduced and in what regions. And so we recognize an opportunity through some sort of public infrastructure to solve for some of those challenges. And so when we thought about the core elements of public infrastructure, we really thought about the physical dimensions, the digital dimensions, the human dimensions, and the integration and sort of stewardship of the entire system or our infrastructure. And so when we think about physical infrastructure, we really were talking about something like what is pictured on the slide, thinking about the range of metacosms to mesocosms and microcosms. So really being able to try to recapitulate the physical environments in which these organisms might be introduced, but also identify that there's an opportunity to think about scaling down and doing some stress testing so that we can fully do a characterization of an organism. So if sort of thinking about a full omics profile of what those organisms are doing and how they're behaving and the response of different environmental signals to that organism. And so there you could start to see already the integration between the physical and the digital, as a lot of those technologies and characterizations would require a data-heavy approach and the ability to then move into thinking about modeling, data analysis, simulations, so that over time we can get to better predictive capabilities to understand how an organism might behave. And so in that digital space, we're starting to see that aggregation of data around what the physical environment allows us to test, but also thinking about that mass aggregation of information around release events. And so this could be in a regional context. You could think individual countries might want to do this, but there's also the potential certainly for a global set of reporting and information aggregation around where different organisms have been released and what was sort of the findings and impacts that we were able to document as a result of those releases. And that would give us more information again around the complexity of how these organisms are behaving in environment, and are they having their intended effect, and how can we really do some like long-term post-market surveillance of some of these introductions, and again in that engineered versus and as well as non-engineered introductions. And then of course there's the human element. Any form of infrastructure is going to be laden with the values of the people who are designing that, but also the people who are impacted by that, where these facilities might exist, how stakeholders would be engaged. Is the facility and are these types of infrastructures really providing the information that is needed for decision making and for building public confidence? And so you could see that opportunity there for also training capability, so that you see that human element woven throughout there. And then really trying to think across all of these different structures, how we might integrate the entire set so that we can have better decisions, better understandings of the how information will sort of flow across the system, and making sure that it is reflecting the values of the people that infrastructure is seeking to serve. So in the conversation, we were a fairly US-centric group, and so we would certainly appreciate input from others with different perspectives and different, I would say, decision-making environments. I think that would strengthen the report out and the report that we're working on. But we also recognize that there's a number of different uses for this type of infrastructure, right? From whether it's informing regulatory decisions, again, building public confidence, training facilities, or just getting a better understanding of how the natural environments and systems are trying to capture and understand what the dynamism within different ecosystems means for our ability to release new technologies or even releasing non-native species into a new environment. So I'll stop there and look forward to the discussion.
Great. Thank you, Andrea. Okay, Zach.
Hi, everybody. I'm Zack Abbott. I'm a microbiologist and CEO of zBiotics, where we make genetically engineered probiotics. And so I was really excited to be part of the conversation that we had, where we really set out to discuss safe design guidelines, the idea of like trying to put together some sort of guidelines that a scientist could use to develop kind of like safe products that were intended for release. But quickly, as we started talking about that, we really struggled with the definition of what safe really meant. And, kind of started by identifying risks as well as benefits. So realizing that risks alone were not really sufficient in kind of talking about a new technology, because everything has some level of risk. And so it was important to think through why you were setting out to do the project and the benefits of the project as well as the risks of not doing anything. And we also quickly realized that as we started trying to define risk and benefits, that fundamentally context for the project was essential. A lot like what John was saying in their discussion as well. And so core design principles were extremely difficult or nearly impossible to design that would be broadly applicable to any project. And so this idea of building a sort of safe design principles was became kind of too daunting to undertake. And so we pivoted that ultimately what we were doing in this conversation was sort of like walking through a series of questions to try and figure out what the risks and benefits of the project would be. And I thought that, or excuse me, and we thought that that developing some kind of worksheet for a scientist to embark on at the design stage before they've sort of picked up a pipette to evaluate the benefits and risks of their project as it's currently designed would be beneficial. And so the opportunity we had here, beyond sort of just a standard sort of like regulatory framework, was that we could kind of cover more ground and be more broad in the considerations that a scientist should be thinking about. And so we built this worksheet, which is pictured here. And so the outline on the left is has an introduction and purpose, and then sort of the key evaluative areas are sort of a benefits analysis, a risk analysis, and then reevaluating your design based on those benefits and risks. Ideally, redesigning or dialing up or changing your design or tweaking your design in some way that could dial up the benefits and dial down potentially identified risks. And so on the right is shown just sort of a screenshot of a small segment of what sort of some of these risk questions might look like. But ultimately, it's important to note that a scientist alone is not qualified, and this is clear in our conversation, not qualified to evaluate all these risks and benefits themselves. Mostly these questions were meant as ways for a scientist to kind of like at the beginning, at the design stage, to identify the things that they need to learn more about, and hopefully speak with experts in relative fields to evaluate the risks and benefits of their project. So they may be an expert in sort of biological or human risk, but as they evaluate sort of environmental risks or legal or social risks, conferring with relative experts to gain more information so that they can really have an informed opinion about the risks and benefits of their project before they ever embark on that project. And so the last thing I'll say is that what we've put together here, we know by no means is comprehensive. We sort of started this process, but we see this as sort of a step one. And a lot of expertise was not represented in sort of the process of building this out. And so we did our best to try and cover as much ground as we could. But as other scientists in other fields of expertise, other experts in sociology and ecology and things like that, take a look at this, and we hope that it can evolve and more and become more. We want it to be dynamic and become basically more useful and more comprehensive and more beneficial to and hopefully be helpful to kind of like scientists as they begin their journey of kind of designing a project intended for release. And with that, I'll pass on to somebody else.
Great. Thanks, Zach. Okay. Yonatan.
Yes. Hi, everybody. I'm Yonatan Chemla. I'm a postdoctoral fellow in the biological engineering department at MIT, where we develop tools and approaches for the safe and effective release of microorganisms into the environment. And I think that is one of the main reasons I was invited to also participate in Asilomar, where myself and a group of very diverse participants from industry, academia, NGOs, and more came together in one of the so-called recombination sessions to discuss and write about a technical science-based report on the risks of beyond containment. And of course, many things were discussed, and then we wrote a very long, relatively long document or summary of it, but I think it could be boiled down into 4 or 5 main points that I want to convey today. First, we recognize that release or beyond containment release is not new. For centuries, we've released beyond containment animals, plants, microbes across ecosystems, sometimes intentionally, sometimes by accident, and in the process, we reshaped entire landscapes and ecosystems. And also, some of these were very beneficial, other cause harm, but it's not new. Also, the release of genetically engineered organism to the environment is not new. First, you can look at large-scale engineering projects like wine fermentation as engineered release. You can look at traditional breeding as release of engineered organism. And also today, we're already releasing like zBiotic engineered organisms to the environment, but also through waste streams of various industries, we're releasing recombinant DNA to the environment in kind of large scale through various processes. So it's not new. What is new is our ability today to engineer them in a much higher precision and speed through advanced genetic engineering. And number two, we also recognize that today that we have a more pressing need to harness these precision and speed that we can engineer to face some of the global challenges that we have today, including widespread pollution, release of greenhouse gases, nitrogen overload, persistent toxic chemicals, and more challenges that could be met if we were able to use genetic engineering to solve these problems. And the third point is that given the first 2 points that I told you that we've already been releasing in large scale for a very long time, and two, that there is a need to enable new innovation to face global challenges, we think that there is kind of an urgent need and a remarkable opportunity to study academically and otherwise the risks, the actual risks behind release. And our key insight was simple, that we need to assess the release of organisms beyond containment based on what they do, not on how they were made, right? And whether engineered or not, released organisms can pose risks to the environment and to human health, right? And in fact, engineered organism, we think, not only that they don't inherently represent a greater risk, we think they could actually be used to reduce risk for beyond biotechnology technologies, with examples of we can engineer kill switches, we can engineer synthetic nutrient dependencies, we can engineer gene flow barriers and that would limit their persistence and spread in the environment. So we think this kind of functional design-based safety is not yet standard or represented in risk assessments. And there are many different risk assessments that we touch on in our report, and we believe that it should be. Fourth, today we need to recognize that we have much more powerful tools at our disposal compared to maybe the original Asilomar 50 years ago, right? We have the ability to do precise gene editing to the level of a denucleotide. We could do very powerful metagenomics, AI-driven modeling, real-time environmental sensors, all of this to allow us to track released organisms, measure their impact, and if needed, we could also sometimes, or we could try to reverse them. So I think these tools bring two new game-changing capabilities. One is that they enable us to start quantifying the risk that a lot of people say that will be very hard, but I think modern technology could help us there. And two, it allows us to start actively mitigating or discussing how we can mitigate that risk. And then this is what we did in our work group. We basically kind of enumerated the different risks that we see in the release of beyond containment technologies. And we went one by one and we discussed what can we measure it with today's tools, and can we mitigate it with today's tools? And then we also discussed specific examples. And you can see the list here on your screen, but I won't go into all of them. I'll just give you three quick examples on the bottom, 10, 11, and 12. So reversibility, I think, is a very important risk assessment tool. Whether or not our beyond containment technology after release could be reversed to the original state or not. And we have tools today, metagenomic and otherwise, to assess the reversibility over time of these environments. And there's several examples. Secondly, comparability. How does these technologies compare to what we're already doing, right? So for example, if I'm already releasing 23 billion liters of fermented microorganism from wine industry a year to the environment, maybe releasing something similar would be comparable to what we've already been doing. And of course, there is a need to assess if what we've already been doing is safe or not. And then whether or not our organisms for release compared to known safe organisms that we use across industries. And then finally, we always need to think about alternative. What is the weight of, we need to realize that there is a cost to also not doing something. What is the cost or the no action alternative of choosing not to release? Does it carry risk or not? Does it creates continual environmental degradation, or it allows pollution to go unchecked, and so on. And finally, just to conclude, so we in the group believe that it's time to maybe embrace a modernized, evidence-based risk assessment approach, one that balances innovation and responsibility, and look at biotechnology not as necessarily a threat or a risk inherently, but maybe as a tool for improving plant or environmental health and human health. Thank you.
Great. Thank you, Jonathan. Okay, Ice.
Hi. Nice to meet everyone. My name is Ice. The full name is Cholpisit Ice Kiattisewee. The name is Thai. I'm originally from Thailand. I'm a postdoctoral fellow at MIT right now, working on synthetic biology. So we engineer organism. And the conversation that I was part of in this working group is mainly the intersection of biotechnology or genetic engineering with the ecology or conservation approach that we are trying to achieve right now. So first of all, what I have to deliver for the whole group is that we don't have a consensus from our group, because there is a different spectrum of a conversation that we've been talking through. There are some different approaches that we touch on, and we decided that we're not going to come with a major statement or consensus from the whole group, but we want to keep this discussion continuous. However, there are some like subgroups that we sort of like organize on, and then we lead into another consensus in treaty and other documents. So there will be 3 treaties in total for our intersection of biotechnology and ecology conservation. So for this one, I'm going to be talking about the reporting out of the overall conversation that happens during the session. So when we start the conversation between the biotechnology and the ecology or conservation angle, the first question or the first few questions that come to our conversation is that, like, how should we use the biotech for the conservation at all? Like, are we allowed, or should we do the engineering or genetic engineering to prevent biodiversity loss? Like, is that a thing? Because if we engineer them to keep the organism around, are they still considered as a conservation? Because they may not be the actual organism that was the one that we are trying to conserve anymore. So the discussion sort of like depends on the viewpoint of people, whether you consider the engineered organism to be something that you're conserving, or whether they are not the same organism anymore and they're another form of living beings that were out there. And like the key conversation that we've been discussing through is that, like, when we're trying to conserve the ecology or like any particular species, is that, like, are we aware of all the consequences that may come afterward? And that's a big thing, because the engineering that we are trying to do, and we're trying to work on the beyond containment setup that we have here, may not be that easy to comprehend on like how far the action will go. So 2 main things that come up in terms of like the consequences is whether, like, are we able to evaluate the outcome between the action and also the inaction? Like we can evaluate the risk of like doing the deployment of this biotechnology in the ecology conservation versus not doing anything. And at the same time, when we do that, there could be some control or some regulation that we should think about when we deploy them across border. So 1 or 2 easy examples that can come up is, like, for example, the coral that has been bleaching all over the world is kind of an urgent issue. And whether doing that or not with different approaches is going to be a big thing. And one conversation that come out is that, like, is biotechnology really the right thing to take action on this? And is it like one of the two or like one of the main two that should be using or not? Because like sometimes the timing of the application should be concerned a lot because it may take some time for us engineers to come up with a proper system to tackle these kind of global issues. And one thing that we came up during a discussion is that like if you think in the conservationist mindset, we think about like a small changes that can make a trophic cascade, like do the small change that can change the whole ecology. But most of the time, the engineers will just try to come up with the optimal designs or optimal genotypes. So the conversation has to be met in order to connect between each other. So since the species are going extinct all the time, we have to be very aware of that. So with that, we have some emerging points to be considered. We want to make sure that we extend connection of the biotech people and the conservation biology people and the social science community to make sure that this conversation continues. We need more risk assessment strategies. And yes, we are trying to connect between our working groups as well. And we have to make sure that the ELSI, the ethical, legal, and societal issues on the long-term ecological impact are well evaluated. And with that, we need to make sure that we address some of these urgency and also the governance beyond, the boundaries of the borders of different nations. We have to be aware of that as well. Last but not least, I want to make sure that I mentioned that like with several news about the de-extinction things going on, we didn't touch too much about the de-extinction because we want to spend more time on the prevention of the biodiversity loss before going to the other stages where like it's already lost and try to make it come back. So I think that would be the overview of our group for the reporting out. Thank you.
Great, thanks, Ice. Okay, Felix.
Hello. I cannot see my cam.
Not yet.
Not yet.
Okay, we can hear you, but we can't see your camera.
Let's see if the host has disabled the option. Okay, I'm sorry, guys. It seems that there is a technical problem there that is not in my hand to solve, but I'm going to continue. So many thanks. Okay, it is already here. Many thanks, Richard, for moderating the session and for sharing this group together with John. Absolutely. And of course, also to the IT team from the Linde Policy Center for making this webinar possible. And I'm glad to present the nature conservation in the age of bioengineering or biotech in treaty that it came from the Asilomar, Spirit of Asilomar Summit, as we know. And just to take a little break, it's just to find out where we are now. No? As you can see in the slide, we are talking just about one of the theme of the Spirit of Asilomar Summit. Within the 5 working groups, we are now in the intersection with ecology and conservation, and there we have at least three outcomes or outputs from this conversation. The first one was the one that my colleague Ice already talked to you guys, and now I'm with the second one on nature conservation in the age of bioengineering. In the next slide, you will see that this treaty is not a consensus document. Absolutely not. It reflects the key reflections, let's say, of a subgroup of participants, capturing some of the opportunities and especially the urgency that we felt during the discussions. And well, we all know that biodiversity loss is happening at an alarming rate. And at the same time, biotech tools are advancing rapidly. And we have all today with this very big news about the dire wolf. This is just an example. So the central question we think that is how we can use these biotechnological tools to help conservation, and how do we do that responsibly. So, and this statement is just, or this entreaty, sorry, is the starting point for answering those questions, or at least for refining those questions, and absolutely not the final answer. In the next slide, you will see the first 4 points that briefly summarize the key reflection of this statement. The first one is the intersection of biotechnology and conservation brings both opportunity and risk, as we already heard. So we need careful, sustained, and case-by-case discussion. The second, biodiversity loss is accelerating. We need to stress this out. The third one is that we come together in the spirit of Asilomar as a multidisciplinary team of experts, and calling for transparent governance, rigorous risk assessment, and broad stakeholder engagement. The fourth point is that collaboration is essential. As conservationists and biotechnologists, we need to work closely, constantly, to ensure that potential interventions are scientifically sound and that are ecologically appropriated, and more important, ethically grounded. In the next slide, you will see the other half, the other 4 points of this entreaty. Is recognizing that yes, we need to be guided by precautionary principle, but we also recognize that inaction or even keeping the status quo could carry serious consequences. And also that biotechnology is not a magic fix, absolutely not. But it can be a useful tool when developed collaboratively and with transparency. And that through open dialogue and interdisciplinary collaboration, we aim to respect nature, honor cultural heritage, and ensure innovation support ecosystems and communities. And finally, this is an open invitation to join this conversation, to contribute with your expertise, and to help shape the future of responsible use of biotechnology in conservation. And finally, in the next slide, just to be clear that this entreaty is not about pushing a single agenda. It is about keeping the space open for fruitful engagement. And I repeat, that is grounded in science and guided by ethics. And yeah, we also see this as a part of a broader call to action, to think together, to act responsibly, and to involve the widest possible set of voice. So thank you so much for listening this very short talk. I am Felix Moronta from the Regulatory Science Group in the International Center for Genetic Engineering and Biotechnology in Italy. And well, yeah, part of my work is focused on helping national and international agencies in understanding how to best regulate the products of emerging biotechnologies.
Great. Thank you, Felix. Okay, and so for the third at this intersection, we come back to Ice.
Hi, I'm here again. So I'm representing a few of us who are trying to push the boundary further for these intersection of biotechnology and conservation in a call of action. So this is like one of the 3 entreaties that came out from the group, and this one is particularly going forward to establishing the global network. So we came up with this acronym GNOME, like Global Network for Organisms and Multidisciplinary Exchange, just to be catchy. But the real intention of for us is that like we are a group of next generation leaders. So the key features of the next generation leaders is that like we attend the session that we are interested in. So like a few of us attend in these like ecology and ecology and biotechnology intersection. But there are a lot of like other people who attend the other sessions, and we see that like one of the key things that we are missing here is to make sure that we have a formal and established network. So in this particular call, like all of the a lot of the participants who are from the NGLs that attend a lot of sections across the Asilomar meeting try to focus on the path forward. So we are trying to make sure that we have some established workflow to support the collaboration and also the participation of the other experts, even beyond the Asilomar conference meetings, and try to make sure that the communities are growing. So we have 3 points coming from these entreaty. We want to make sure that we build and strengthen the trust. We want to make sure that the communities consist of both scientists, ethicists, and other stakeholders. And we want to make sure that we include the global stakeholders, inclusive of the global South community. Two, we want to make sure that we foster the exchange of the technical and the cultural values, including the local knowledge and perspectives, just to make sure that we can inform all the decision making that is involving the ecology and the biotechnology frameworks properly. Now, we believe that the cultural values are very important to move forward. And three is to make sure that we build responsible and accountable biotech while we respect the nature. Like a lot of times, a human has been exploiting the nature or the natural resources for a relatively long time. I think we can see a lot of changes going on. And now we want to make sure that, like, while we're moving forward with these missions, we want to make sure that we keep our respect for the nature. We respect the local community values when we deploy anything at the local community, and we want to emphasize the needs and rights for each community to move forward. And with this, when we are establishing the network, we want to make sure that we invite all of you to join our networks beyond the Asilomar meeting and make sure that we formulate these initiatives together.
Great. Thank you, Ice. Okay, and finally, we'll hear from Emma.
Hi, everyone. My name is Emma Frow. I'm based at Arizona State University. The job of trying to pull out a few kind of common themes from across the panel remarks that we've just heard. So I think what I'll do is move sort of in reverse order through the reflections, and start by saying that my sense is that we've just moved through several different topics, but underlying them, there's a lot in common. First, I think across our panelists, there's a shared sense that biotechnologies beyond conventional containment is a really challenging topic. And I think it's challenging in part because what's at stake goes beyond science and technology and raises bigger philosophical and ethical questions about our relationship with the world today, right? So, for example, the question of whether we should use engineered organisms to support conservation gets really deep into how we conceptualize our responsibility to the natural world, and what kinds of approaches we think are acceptable to try and support ecosystems or to remedy damage, like to coral reefs and things like that. So, and not surprisingly, answers to this might vary across cultures and borders. And I think these questions go beyond conservation to sectors like agriculture too, right? So, for example, what kind of world are we promoting when we engineer crops for higher yields in ways that lead to increased industrialization and fertilizer use? And how do we start to balance the environmental and food security considerations around agricultural biotechnology, right? So not just sort of thinking about risks in isolation, but how we balance things. I think too, the number of reports that we've just heard that have mentioned that these ideas apply to non-engineered organisms as well as engineered ones reinforce to me that these questions are bigger than just biotechnology, right? So we have experience. We know that introducing new species, new organisms into environments can have positive effects as well as negative consequences. So I think in part what we're grappling with here is not specific to biotechnology, but to how we approach the whole idea of biological introductions and managing our natural world and our kind of engineered natural world as well. So I think across most of the remarks today, we've heard several calls in different ways for processes and infrastructure that would allow us to work through the web of possible interactions and consequences, and sort of looking for guidance for how to make clear decisions about whether introductions would be net beneficial or not, whatever beneficial means, which is something complex to unpack too. So I think different parts of the Spirit of Asilomar discussions have been trying to map out at various levels what this kind of decision-making infrastructure would look like, right? So we've heard report outs just now that include sort of deep dives into research design and testing and calls for kind of monitoring and tracing capabilities, and several times too a desire to engage more broadly across public and stakeholder groups. And I think all of these to me are about how we sort of design our infrastructures to support sound decision-making in complex and uncertain environments. So across all the remarks, I think we've heard about a number of different elements that feel important to attend to. I don't think we've covered the full list, so it would be great with our discussion time to have conversation that helps us to start fleshing out these elements more fully, and especially to think about them at different geopolitical scales as well. And then I think to sort of go right back to the opening remarks in the webinar by, I think, both John and Richard sort of talked about this kind of move. Richard characterized it as like the move from factory to farm to field, and how it gets harder to conceptualize containment there. So I think, just to sort of finish with this kind of big picture question of saying that, like, it feels like our 50-year-old concept of containment in biotechnology really revolves around trying to separate technology from society. And my sense is that as a group, we're starting to collectively ask whether that's the best starting point for the conversations that we're having today. So is separation necessarily the goal or the only goal when we're talking about bioremediation efforts or organisms that are designed to persist in the environment, right? If these technologies are out in the world, should our conceptual framework be grounded not in a mindset of separation, but of assuming entanglement and thinking about how we hold this bigger, more open system together, and thinking about kind of the complex set of both risks and benefits to environments, but also the kind of complex socioeconomic systems we have today. So my sense is that this is the kind of territory that we've collectively been starting to like map out and work through in the Spirit of Asilomar discussions, and that this feels like a really timely opportunity to continue building a highly interdisciplinary, multi-stakeholder discussion around this. So I'll stop there. Richard, I don't know if there's more slides, or are we moving straight to discussion?
We got one more slide, which is just to say that we're going to move to discussion. And so what we'd like to do next is, have a discussion and get some of your questions. There's a Q&A button that you should see on your Zoom link. And if you put your questions there, then everybody in the participant list can see those questions. And you can actually upvote those questions and sort of pick the ones that you would like to see asked first. And so I would encourage you to do that. That kind of helps us get a little bit of a sense about what people would like to ask. I would also, while you're thinking about that, like to point out that in addition to this webinar, we're going to try a little experiment, and we hope people would be interested in participating, which is to have some office hours. I'm not sure that's the best term, but basically an opportunity to have an informal, unrecorded, come in and just chat with us about any of the areas that you've heard. And so we've set aside 4 different slots next week, and we'll email these out to you on the mailing list. If you signed up for the webinar, you will get this in email also. But next week on Monday, Tuesday, and a couple of them on Friday at different times that we hope will accommodate various people in different parts of the world. Collection of 4 to 5 of us will be available to just talk about things and get your input. And as you heard, we're putting together these entreaties. The timing is that we have to do our updates to the entreaties soon. And so we get the feedback coming up by Monday from everybody. This basically provides a way in the Beyond Containment theme to get feedback from people who weren't at the summit as well. And so we hope that you might provide some of that feedback. So, and there's a Zoom link there. And again, we'll send all of this out. You can, screenshot this if you like, but we'll send all of this out to you. All right. So with that, let me stop sharing my slides so that we can kind of see all of us. And let me ask all of the panelists to kind of turn your video on so we can see everybody.
Just need the host to enable me to do that.
Okay.
This is Zach. Yeah.
How you'll do that in a second. Great. And we got a couple of questions. And so let me start with one maybe addressed to everybody about activities in Europe that would be applicable in the US. And maybe I'll start with Felix, since you're sitting in Europe, and you can say a little bit about that. But then I might also ask Andrea to comment, because I know you've done some things with the UK specifically and might have a little bit there, and anybody else who wants to comment as well. Felix, what about things that are going on in Europe? How does this compare to discussions in Europe? How can we take advantage of those?
Oh, yes, Richard, if someone can please enable my cam.
Yep.
So we are equally.
Pow, if you can take care of that.
Yeah, thank you so much.
Go ahead and start talking. We'll get you there.
Okay. Yeah, now it's better now. Just to clarify, I am based in Europe, but the scope of my organization is global. We are an intergovernmental organization. So regardless that we are based in Europe, we are not that much involved in the regulatory aspect of the region. and so on. So, but as you may know, no, here in Europe, the regulatory situation is particular because it is an harmonization of from a continental perspective. So the bureaucracy that is involved is important. And in the case of this kind of technologies, I mean, we have seen some advancement lately, as particularly in agriculture with the use of gene-edited crops, and researchers saw that part as positive, but there is still a lot of work to do here in Europe to harmonize with other part of the world. Yeah.
And Andrea, I know you were involved a little bit in funding some things that through Schmidt Sciences that kind of cut across Europe. I don't know if you have anything you want to say about, just the extent to which you see differences and commonalities and other things.
Yes, but not through our funding program. I think
Okay.
through different activities and engagements, we did see over the last 2 years, particularly in the UK, an interest in understanding and developing regulatory sandboxes that would help us understand more about how we approach the regulatory decision-making process, and trial and test new ways of approaching those regulatory pathways and the review. Yet that I think there's an opportunity for folks to interpret that as like physical infrastructure sandbox, like an actual sandbox where you test and tinker with things, but that was really focused on new processes for getting to decision making as opposed to physical infrastructure type of testing. So I think there's certainly room for us to learn more about what there is an interest in doing so in terms of the physical testing and ability to get toward information around different release environments. And I'm excited to go through the documents that are being shared by the participants and think about how we integrate some of that feedback into our existing report out.
Great. Comments from others about, sort of things going on in Europe or other parts of the world that we might take advantage of? Okay. So we have another, maybe a comment, but I'll, Andrea, I'm going to address this one at you as well, about some of the past work that's been done in creating public infrastructure, and how this compares, or how what you're kind of proposing compares. What's already been done, what's out there, how does what you're proposing, in your entreaty compare to that?
Yeah, I think there have been field site testing locations that have been created or made available to folks. There has been sort of, as you were getting at with the sort of the moderate release, the farm environment where we understand buffer zones and different, there have been those types of structures and locations that have been created. But I think what we are talking about a little bit here was something that is, at its full extent, that huge metacosm is much grander in scale. And would also still be fully contained, so that we're not actually testing in this in a space that would have the potential to get beyond any type of buffer zone, so that we can also then experiment with different technologies that might be harder to track or pull back in these types of fully enclosed settings. But I think we had a recognition that there is this would be incredibly expensive, challenging to replicate for all the different parts of the world and all the different types of regions and environments and ecosystems, and the connection across different types of ecosystems. Right? Is this upstream? Are we trying to model something that is upstream from a river or a water body, and that might have then either political sort of boundaries that are being crossed, geographical boundaries that are being crossed. So we recognize that there's really sort of a lofty vision for how that might come together, but there would be potential benefit. And that's why also within the entreaty, we point toward the ability to how do we sort of downscale and stress test through molecular characterization of the different organisms and their reactions in different environmental conditions, that we can get towards simulations, recognizing that we're not going to be able to build these types of metacosms and microcosms that recapitulate everything, but really trying to think about what that would look like and how that might work. And I would invite any of the other participants on the panel to also comment on that.
Comments from others? Okay. We have a question here about how scientists should assess the potential ecological harm of specific genetic parts in their released synthetic organism. For example, Cas9, antibiotic selection markers, unnatural amino acids, things of that sort. What portion of the responsibility for exploration of specific parts is on the individual versus on communal and governmental guidance? Maybe we can start with Zack on that one. It seems to lie fairly clearly in the domain that you've been thinking a little bit about.
Yeah, it's a great question. It's one, and that's exactly kind of what we were grappling with in our discussions. And I think that part of what we came to, as I said in my sort of intro slides, was that context it really matters deeply. So like kind of like what gene or what function you're introducing, or what organism you're introducing into what ecosystem, how much you plan to introduce, sort of when you introduce that, how frequently, things like that. So I think that understanding the function of the gene and the presence of that gene in the ecosystem, for instance, thought a lot about sort of, I think a lot of times we equate knowns and unknowns with risks, but in reality, an unknown is just an unknown. It may not actually pose a risk. But the fact is, if something is known, then there is at least some amount of data to potentially evaluate the impact or risk of that. And so if a gene exists in the ecosystem, how frequently does it exist in that ecosystem, and what is the impact of that? And then potentially we can better predict what the outcome of that would be. And so to your question about like who should be evaluating that, I think that it's first and foremost responsibility of the scientist who's endeavoring to kind of like on the project. And we sort of focused on the point in time at which it's before pipette has hit tube, and you're really in the design stage. And so ideally you're responsible for evaluating what kind of the impact might be. And we really encouraged scientists to not do that in isolation, to ideally get help and expertise from those that are equipped to evaluate those. So if there's an ecological question, talking with ecologists or looking into some of existing ecological research and identifying ideally the knowns and the known risks, but then also being transparent and honest about the unknowns, that like we don't actually know what's going to happen here. And so then sort of predicting potential outcomes or looking for ways to gather data to inform what that risk might be. And so I think ultimately it has to be driven by the scientist who's designing the project, because nobody else is incentivized to do it. But hopefully that scientist, and the purpose of kind of our worksheet was around the idea of like helping that scientist think through all the potential outcomes, and reaching out to stakeholders and experts that can help with kind of identifying that.
Great. Thank you, Zack. Other comments on sort of specific components and who ought to be responsible? Anybody wants to make?
Yeah.
Nice.
I want to add one thing that like one of the angle that we can do that was beyond the scientist responsibility is the surveillance. So we touch on that a bit, but like it's not as clear to all of us on like who is in charge of that. Like if we think about the environmental like chemical this distribution, it could be like EPA in the US and something like that. But like for the DNA is kind of harder, because like if you think about, we deploy something that could be like engineered or not engineered to the system. Can we make sure that the thing that we observe from the surveillance approach that we are doing is actually from our engineering approaches? Like, is it true that it's from our stuff, or is it like possibly coming from the other thing? So like there are more guidelines surrounding that area that the discussion is still continued, as far as like we have been discussed through at the conference.
Great. So I'm going to aim this one at Emma and Yonatan. Several panelists discussed the importance of considering the consequences of introductions of all types of organisms, regardless of whether they are engineered. How do we do that? Do we need new law in the US, for example, to provide regulators with that type of authority? Is a case-by-case regulatory paradigm, how can organisms, engineered or not, be rapidly triaged so as not to slow down the release of very low-risk organisms? Maybe we'll start with Emma and then Yonatan.
Sure. Well, I don't have huge regulatory expertise, but I'm tempted to say that the sort of case-by-case product-based approach is one that, at a base level could maybe, allow for this kind of work, Becky, like in the sense that thinking about not necessarily the process, but the sort of the function of the product, the novelty of the product, and so on, whether or not it's genetically engineered might allow for that, right? So, I think, part of me thinks that like the default setting is not necessarily one that is deeply problematic that way. But I think what we do need is sort of better sets of questions and guidance to help do that triaging, right? To sort of understand what it is that we're looking for, especially when we're talking about sort of outside the farm types of releases into more complex environments, is what are the triage questions that might be used, right, to make those evaluations. So that this may not be a wholesale reworking, but that we need to kind of really flesh out and develop and think through coordinated processes for that element of the regulatory process.
Great. So maybe Yonatan, and I'll ask John to comment on this one as well.
Yeah, thank you. First, maybe I'll go back to your first question, Becky, and I'm not sure if we discussed it in our session or not, because I remember you were there. But the question of when do you go to the government is actually a very important question, because we know that any regulatory burden is actually incredibly inhibitory for innovation, or at least that's what effectively happens in this case. And I do think there are clear cases where there need to be an input from the community and the government, right? For example, pathogens or select agent lists, or questions around the scale of release, right? Maybe a small scale is completely different than a experimental small scale release, etc. But I think currently the regulations where we need or don't need to go to the government doesn't make much sense. For example, the blanket or effectively a blanket ban on genetic engineering is it just simply doesn't always make scientific sense. Maybe there's other values involved here, but at least scientifically, I don't think it makes much sense. So maybe we need to rebalance when we need to get approval from the regulators and we don't need. And then your second part of your first question was the different components of our engineered. So I think a very basic question that you could ask, for example, are these components already in nature? And for all the list, except for natural amino acids that you gave before, are actually already in nature, probably already in the soil you would want to do your experiment in the field, for example. And the same is true for the host organism that you want to release. It might be a native organism to where you want to release. And then there is other question around like the scale of release, how would that affect the steady state of the community, etc. But let's put it aside. And then maybe a last sentence about the other question. I do think that questions should be, again, like I said, not about how the organism was made, but what does it do, right? And what are the risks? And fast triage that you refer to, I think it needs to be more around the risk of spread and the scale of release, and less about, I don't know, the specific method used to release it.
Including no method, right? It's a natural organism because, you had sort of mentioned, right? There's
Yes, exactly.
And there is this question, John, I'll ask you to comment on in a second also, but many US regulations, and I'm not, as familiar with ones in other parts of the world, but maybe the same, regulate things like new chemicals, like TSCA, the Toxic Substances Control Act, regulates new chemicals, right? So they say, well, what's a new chemical? Well, a natural organism, right, isn't a new chemical, right? An engineered organism can be. There are various, kind of just nuances there. But that may require a different statutory authority if one wanted to say, hey, you're taking a natural organism and putting it somewhere. Well, under what authority, right, is that being regulated? John, I know this has kind of come up a little bit in conversations you've been involved with. I don't know if it came up in the ESL one, but any thoughts on this one?
Yeah, no, I think the answers so far have really kind of done a lot of justice to the complexity of the challenge. One thing I'll add is that I think in order to kind of streamline the regulatory process and do fast triage of things entering into the regulatory process, it can be really useful to have a framework that can say, well, this thing coming in is like a previous thing that already went through the process, whether it was approved or disapproved or things like that. And I think these sorts of evaluation frameworks can be really useful in order to provide a basis for kind of saying, what does it mean to say this thing is like another, right? With biotechnologies beyond conventional containment, they're very challenging because it's not just the nature of the biological agent and the nature of the engineering that makes up what the product is. It's also, in addition to those things, the nature of the use case, intended and also unintended. And so we have this kind of dual nature of what a singular product is that makes it even more challenging to ask the question, this thing is like another thing. But that doesn't mean it's impossible. And I think some of the most important efforts and impacts coming out of the efforts to generate these evaluative frameworks will be the ability to kind of, at least as a first pass, make these sorts of assessments so that kind of on broad strokes, you can kind of say, okay, this is like that. Let's now move forward and do more specific tests regarding the nuances of the properties by which maybe there are some dissimilarities, but broadly they're similar. So yeah, I think that kind of example really highlights why this work is important.
Richard, can I just jump in and say one thing?
Yeah, let's do Zack then Emma.
One additional thing I'll just say is that, like, one of the challenges with regulatory frameworks is that in regulation and looking at the government is that by necessity and by definition, they're rigid and they're not dynamic. And that can prevent a challenge. And so, when you're talking about case by case basis things, this is not where regulatory frameworks are set up for. And so I think this is a role where sort of self-regulation or industry regulation becomes really important and really vital. And that triage can kind of happen before it kind of hits government regulation and allows for some more of that rational kind of dynamic case by case as things evolve and change, because regulations change so much more slowly. So I think that ultimately that's where these roles I think have to be different.
Emma?
Just thinking a couple things that, as we're continuing to flesh out this work could be interesting is looking at, for example, like in medical devices, there are pathways, for exemptions based on similarity to other products and whatnot. It could be worth us looking into the kinds of ways in which those have and haven't worked to understand how those might be applicable. But also just to sort of put out there a bigger question when we're talking about low risk and kind of making assessments about low risk is, I guess, a question for us to think through is what are the parameters of risk that we're talking about? So is it just risk to environmental and human health and safety, or are we thinking about risk in broader terms as well? Socioeconomic risk, geopolitical risk, things like that as well. And how do we factor those into decision making around the introduction of new species as well?
Great. Okay, we have another question. I'm going to throw this open to anybody who wants to answer it about as biotechnologies continue to advance, particularly in gene editing and synthetic biology, how can we ensure that the regulatory frameworks around containment evolve to not only manage biosecurity risks, but also promote responsible innovation, especially in the context of open source biotechnology and decentralized biohacking communities? Anybody want to take a cut at that one? Yeah, Felix.
Yeah, thank you, Richard. Yeah, that's a very good point, and this is the a constant question, that we all always have in mind, how to find the right balance between promoting innovation and focusing so much on the risk. And I think that, well, in the case that the person, I don't get the name, the person is asking on open sources and other kind of approaches. I think that the important part there is about the trust building. There are mechanisms for streamlining the biosecurity or safety, let's say safety aspect within a research project, and this is that's the way it should be done ideally. One example the first example that comes to my mind is the iGEM competition. So it is very pushing for innovation, absolutely, but at the same time, the safety issues are transversal in all the projects in whatever initiative that is there. So it builds not only in the innovation, but also in safety, ethics, and security reviews. And it should be hand by hand, absolutely, not to see them like confronting aspect, but just to complementing aspect. the perfect and definitive answer, well, it depends of the context, the jurisdiction, the values, etc. But I think that the key is trust and is collaboration between the two perspectives, I think.
I'll say one other thing on this one, and others can join in as well. In the DIY bio community, there's been a lot of activity over the last couple of decades and really developing community norms and thinking about these things. And there's sort of a nice shared governance that I really am impressed with in the DIY bio community, where they've thought about these things. And they, I think this gets discussed. You go to a DIY bio lab, and I think one of the first things they talk about is, well, let's talk about what you're doing here in the lab, and let's think about the ethical considerations, the safety considerations, and other things. And so having those sort of shared community norms is certainly one thing that can help there. And again, the DIY bio community, just because it's mentioned, has done a great job at that, in the past. Another mechanism, similarly for kind of sharing those norms, is through things like iGEM, right, where there's a lot of effort. So iGEM, if you don't know, is the International Genetically Engineered Machines Competition. Involves college students, but also high school students and others from around the world. It's a huge activity, and there's a lot of activity there about having the conversation around these considerations around regulatory frameworks and security risks and ethical risks and other things. Other comments on this, Emma?
Yeah, so I think absolutely to echo I think what Felix and Richard were just saying about, I think questions of culture and stewardship are really important to kind of grow within the communities. I think what Zack presented in his remarks with the sort of really specific questions and guidelines and things like that are also really helpful tools, right? To have kind of openly available for people to kind of wrestle with as they're thinking about technologies. I think too, and maybe this gets at one of the other questions in the chat, I think having clearer responsibility and accountability mechanisms when things go wrong also becomes really important, right? So really kind of making sure that our governance structures kind of cover not just the upstream, but really thinking about sort of downstream, how that plays out. There might be some interesting international kind of differences and approaches we can think about there as well. And I think also as a group for us, making sure that sort of infrastructure that gets developed is accessible to players of different sizes, right? So that like smaller groups can readily plug into infrastructure for, for testing, for engagement, and so on. Not just necessarily having infrastructure that kind of is more or solely accessible by larger players, right? Like that's all part of sort of building this culture of stewardship and responsibility.
Yeah. I'd love to just kind of like build on that. I totally agree. And I think you made like some really good points to underline there are this. So I think zooming in on kind of that like, how do we ensure that regulatory frameworks, that question, I think sort of presumes that regulation is the way that we develop responsibility. And I think that for instance, in our working group where we talked about building this sort of like worksheet that at the design stage is not regulation. And I think that more and more the community builds infrastructure, as you said, Emma, that people can access. Sometimes regulation actually benefits the biggest players, right? Because it now becomes a barrier or a moat that new innovation can't reach. And so it's very important, I think, to build a community of responsibility and allow, I think at the end of the day, when people embark on a project, the vast majority of the time, they want to create good. So let's give them the tools to evaluate their impact and sort of like create a community of stewardship and responsibility. And I think that's how you build a safe sandbox for innovation that also that takes into account responsibility. And then regulations can follow on later. But ultimately, I think if we look to regulation to solve all of our problems about responsibility and innovation, we are definitely going to fail.
Andrea, you want to say something?
Yeah, I just want to add on also thinking about some of the existing infrastructure that we can build on, right? So we have historically had a lot of ethics classes within graduate schools that focus on plagiarism and scientific integrity. Could those be expanded to consider from day one as folks are learning the tools of the trade, the tools of biotechnology, not just in a biomedical context, but in any type of field where they're going to learn advanced tools about molecular biology? Should they be learning and could they be learning learning in those sorts of classes that already exist about regulatory pathways, about what biosecurity is, about responsible governance, so that you can start to build that broader awareness of the ecosystem into which their scientific developments are going to enter into. And so that we can then have a more informed community about all the different structures that regulate softly or formally our work, but then that also then get that information would then also blend into their engagements with DIY communities and sort of community laboratories. That you'll have many more people who have a better sense of what the existing structures are to then engage with others who are outside of that, who then might be well informed in some future, think about what those new structures can be and how those structures can start to evolve. But I think we don't formally teach those things in our existing schools, and that might be an easy way to build on existing infrastructure to start to see those pathways for evolution.
Right. So I have one that kind of falls a little bit from that, which is, how do we include the right people in these conversations about what's going on? So there's a question about which specific stakeholders do you think are essential to include in conversations around biotechnologies beyond conventional containment? I'm interested to hear your thoughts, especially as to how to include people that historically have been excluded into these governance frameworks. Ice, I know there was a little bit in some of the NGL stuff about that. Maybe I can ask you to kick that one off.
Yeah, they have we have a few in the call as well who are here that might be able to answer better than me. But I'll rephrase what I've been the conversation I'll be talking with them. So it is very important to make sure that when we're deploying any biotechnologies into the actual communities to include the people inside the community who will be directly affected by the technology deployment into the conversation. So I know this is going to be hard, right? Because like if you're deploying to like 20 different sites in the world, like how can you like bring 20 group of people into the same conversation in, a kind of like global meeting? But at least if we can start from like bringing in representatives or bring in people who understand the context or people who have similar background who can share what is the cultural values or the key interests of those group of people into the conversation. That would be the first starting point. So I know that like there's a lot of like intersection between a lot of international organizations led by UN, UNESCO, CBD. A lot of these conversation has been going on already, and would be good for the US and other communities to follow and like bring in more stakeholders from the actual deployment sites into the conversation as well.
Any other comments on this one in terms of how to include different groups, Emma?
I think one thing too is to kind of push for the idea, especially if we're thinking about, technologies, organisms designed for use in the world, is to really design alongside stakeholders. So not just design and then and then make sure that the right people are okay with it, but kind of really thinking through the kind of practice of technology development for these stakeholders as well, and making sure that, depending, and this is where the question of specific stakeholders is really a case-by-case question, I think. And really involves understanding, the use, like the possible sort of use or intent of the technology, whether it's something that different stakeholders have the choice to opt in or out of, for example, and then figuring out, at different kind of levels, different degrees of separation, who is likely to be affected and how, right? And really trying to map that out. So I think, I think it's a really important question, and it's one where we need to kind of think through how infrastructure is able to make sure that key perspectives from individuals who are either directly invested or who are going to be impacted, even though they're not directly buying in, are incorporated into not just the deployment phase, but actually deep into the design of the technology itself, or the conception of what the problem is and how best to address it.
Yeah, just to build on that, I like what you said about the idea of not just designing and then asking for approval from everybody, but also like incorporating those conversations into the design. And part of the idea of the worksheet we built was exactly that it raises these questions of who are the stakeholders. And importantly, I think an important concept as well is, are the same people who are going to benefit also the ones who are going to be bearing the burden of risk of any new technology, and making sure that those things align. And so I think at the design stage, you can ask those questions. You can ideally kind of have those in a best case scenario, you're having those conversations with those stakeholders, and ensuring that your design aligns basically benefits and risks with the same communities. I think an important thing to keep in mind, and another thing that we grappled with, was this idea of like ensuring that, like, for instance, we don't have stakeholder conversations when we deploy most technologies, right? Like, we don't ask the community when a car company puts a new bit of technology into their car. And so making sure that we're being consistent in thinking about how these technologies, the unique risks or challenges are addressed, but that this idea of putting a unique burden on biotechnology it should, I think, just keeping that concept in mind, that this is technology. Like any technology, all technology presents risks and benefits.
Great, thank you. Okay, well, we've sort of reached the end of our time. We're not out of questions yet. There's still more, but I'd like to remind everybody that there will be more opportunity. Well, first, thank you all for participating in terms of the participants and some great questions. We'd love to have you participate next week as we have our office hours. Again, we'll send those out via email. Also, if you would like to be put on the mailing list for future activities, I've just posted in the chat. You can send email to Sarah Kassir and she'll put you on the mailing list to hear about future activities related to this. But we'll send everybody who was on the webinar a link to the information for next week. And it'd be great, if you're interested in saying more and having more interaction, these will be informal. Just, come, show up. We'll have 4 or 5 of us at each one to talk with you about things that you heard today and other things that you're interested in. Share more information with us. This will help us strengthen our entreaties. So thank you all for your participation. Thank you to the panelists for taking the time to share some of your experiences at Asilomar and some of the stuff you're working on. And with that, we will bring our webinar to a close. So thank you very much, everybody.